Okay, so let's take a look at, let's see if I can do 3 questions at one time, because these are, or 4 questions even, because they all kind of go together. And I'm talking about questions 1021 to 1024. Okay, so these are mostly talking about DNA denaturation and the hyperchromic effect. So let's just start by going over a little bit about DNA. So let's see. So remember in class I diagrammed something like this. So here we'll have a double-stranded DNA, and over here I'm going to draw that DNA as single strands. So this is single-stranded DNA, and this is double-stranded DNA. Okay, so if I had some double-stranded DNA in some water and I wanted to take apart the strands, I wanted to separate those strands, that would be called denaturation. If I wanted to denature the DNA, I would be separating the strands there. So denaturation. And so we could use chemicals to do that. And what we're doing is we're just breaking those hydrogen bonds. So we're not breaking the covalent bonds, but we're breaking the hydrogen bonds, and we're separating those 2 strands. And the easiest way to do that is to use heat. So we can just get some boiling water, and we can put our tube of DNA in the boiling water, and that'll heat it up, and those strands will separate. But we can also do it more carefully, we use like a more scientific method, and we can like raise the temperature slowly. And when we do that, let's see here, we have let's start at like 60 degrees, and I'm going to go up by 5 temperatures, and we're working with Celsius here, because this is science, right? So we're going to work with Celsius. So, and if we raise the temperature slowly, what we should start to see, is that, you know, the DNA will start to denature. And we'll see something like this. So this would be, say, the amount of denatured DNA here. And so at this point, say at like 70 degrees, like, okay, some of the DNA will be denatured, and most of it will still be double-stranded. At here, most of it's denatured, most of it's single-stranded, and only a little bit of it is double-stranded. And at this point, right in the middle, let's say this y-axis is the percentage of double-stranded DNA versus single-stranded DNA. So at this point in the middle between these two, now this point is the point at which half of the DNA is double-stranded, and half, if you know half of the DNA is double-stranded, the other half should be single-stranded. So, and in this case, the way I've drawn this here, this temperature, this 75.5 degrees Celsius, that would be called the TM. It doesn't look like an M, does it? The TM of that solution of DNA, or that DNA molecule there. Let's say I have a million of these DNA molecules in a solution of water. Well, the TM of the DNA there, that would be 75.5 degrees Celsius. So we have to heat that little solution with that DNA up to 75.5 degrees Celsius, and half of it will be double-stranded, half single-stranded. And how do we know this? So this is typically measured with UV light. So DNA happens to be very good at absorbing UV light with a wavelength of 260 nanometers. So light with a wavelength of 260 nanometers, but that happens to be in the UV range of light. So DNA is really good at absorbing light with this wavelength. And single-stranded DNA is even better than double-stranded DNA at absorbing this. So if we were to measure the ability of that solution of DNA to absorb UV light, we would see it going up, like I've drawn here. We would see this going up as we heat the solution, because the DNA is becoming more single-stranded. And again, at this point, this point right here, right between the high level here and this lowest level here, this is an inflection point maybe. This is where 50% is double-stranded and 50% is single-stranded. This is called the hyperchromic shift. And that is the measurement of the hyperchromic effect. And the hyperchromic effect is just the phenomenon where double-stranded DNA absorbs UV light less well than single-stranded DNA. So that's the hyperchromic effect. Double-stranded DNA absorbs UV light less well than single-stranded DNA. And the hyperchromic shift is just the measurement of the hyperchromic effect. Now, so if we took a different solution of DNA here, what if we took a different solution of DNA? So, okay, what if we took a different solution? So this is a completely different DNA molecule. You know, millions of molecules of the same type in a tube. It doesn't have to be the same type, but let's just say it's the same type in a tube. And we did the same experiment here. So this is an experiment we could do where we incubate that DNA at certain temperatures, and then we record the absorbance of UV light. How well does it absorb that light? And we have this DNA here, and we see, oh, it's following a slightly different, you know, similar shape curve, but you know, this sort of this inflection point here, right here, where, you know, it goes from like going up rapidly to you know, less to going up less rapidly. So this point right here, you know, it's shifted over. And what's going on with that? So if we can measure this, so this is the TM of this other solution of DNA. This other DNA molecule has a higher TM, and this one's about, what, 79 degrees Celsius? Okay, so why is that higher than this other one over here? We talked about this in class, right? So the most likely explanation is that it has a higher percentage of GC base pairs, right? So when I can draw this out for you. So let's say this tube in red here, this DNA in red, was like, one strand was like all G's, if that was, you know, possible to get a long strand of all G's. And this one over here is all C's. So, okay, so this one right here, you know, we can put all the bonds in here. Each one of these base pairs has 3 hydrogen bonds between it. And let's go over here. Let's put in the same number of nucleotides. Okay, now each of these only has 2 hydrogen bonds. So to separate, you know, these 2 pieces of DNA here, you know, we can count, let's count up the bonds here. 2, 4, 6, 8, 10, 12, 14, 16, 18. And let's count them up here. 3, 6, 9, 12, 15, 18, 21, 24, 27. Okay, so we have 27 hydrogen bonds there, 18 hydrogen bonds here. This one up here is going to need a lot more energy to denature than this DNA molecule down here, right? Because, you know, hydrogen bonds are weak, but there's just more of them up here than there are down here. So we're going to need more energy, and that energy is coming from the temperature of the water, of the solution that the DNA is in. So that's why this would have a higher TM than this one. And, you know, I've drawn it here where all the bases are identical in these strands, but it doesn't have to be. So any DNA molecule that has a higher percentage of GC should have a higher TM. So higher TM than a molecule that has a higher percentage of, I don't know how I'm drawing that here. So DNA molecule with higher percentage of GC should have a higher TM than a molecule with a lower percentage of G's and C's. So let's take a look at what else we need to cover to understand this group of questions. No, I think we got everything. So now let's take a look. So for 21, what is the physical state of DNA following denaturation? Let's take a look. So we went over that. So DNA is typically double-stranded, right? But when we denature it, it is single-stranded. And we lost my other strand here. Here it is. It's buried under all those lines. But okay, so what is the physical state? So it goes from being double-stranded to being single-stranded. Okay, so that's what we're looking for the answer to 1021. And for 1022, what is the hyperchromic effect? How is it measured? So we talked about that. So the hyperchromic effect is the tendency of double-stranded DNA to absorb UV light less well than single-stranded DNA. How is it measured? And we have the little experiment here to show how it's measured, right? So we just take our DNA in a solution of water and we heat it at different temperatures from, say, about 60 degrees all the way up to about 100 degrees, and we measure its ability to absorb the UV light at each temperature. And then we just look at the difference here, and that's the hyperchromic effect, and that's actually the difference is the hyperchromic shift. And what does Tm imply? Okay, and we talked about that. So Tm is the temperature at which half of the DNA molecules in a solution are double-stranded and half are single-stranded. So let's see, can we do 24? I think we can. I think 24 is a molecular hybridization question. Let me just pull that up. Where is it? So what is the chemical basis of molecular hybridization? Okay, so let's take a quick look at that one. So what I'm going to do here is kind of just go over it like I went over in class. So if we have, say, a single-stranded DNA here, and we have this, say, attached to a microscope slide. So we have some techniques. We can take a cell, attach the cell to a microscope slide. We can make the membranes permeable, permeabilize them, and it can remove some of the proteins and stuff and get that out of the way. And then we can use chemicals to denature the DNA, so we don't have to just heat them to 100 degrees or something to denature all the DNA. We can use chemicals. And let's say we have a single strand here, and to make things easy, I'm just going to make these all adenines. So they're all adenines. And this is say this is a section of our microscope slide. And obviously we're really zoomed in here, right? So, okay, here's our real slide up here. And we're just looking at a tiny, tiny point right there. I'm zooming in. Okay, so we're really zoomed in there, and we're looking at this DNA molecule. Now, if we had a probe, we could make the probe in the lab, or we can order the probe from a company, a biotechnology company. Hopefully it's not too expensive. But if this was a probe and we wanted to detect DNA, single-stranded DNAs that had a bunch of adenines on them, well, we would order a probe that had a bunch of thymines. Like that. And we'd also, we'd want a fluorescent molecule on there too. Now, so molecular hybridization essentially is saying, so what we're going to do is we're going to put this in a solution in a buffer, with a couple other things in the buffer. And what we're going to do is we're going to overlay that buffer on with the probe. This is called the probe. We're going to overlay this on the slide, and this is going to float around, and eventually, because these are attracted to one another. And why are they attracted to one another? Because the A here matches this A, and okay, so they'll be attracted to each other because they complement, and this one will be attracted to that, and this to that, and so on and so forth. So anywhere that we have lots of A's stuck on this slide, this probe is going to hybridize or hybridize to the DNA, form a double-stranded DNA molecule because these bases are complementary. So essentially anywhere on that slide we have long stretches of adenines in a row, the probe is going to stick there. And then when we shine a laser on this microscope slide of a certain wavelength, or just use some UV light or something like that, whatever we need to make this fluorescent molecule actually emit photons and fluoresce in a way that we can detect them. When we do that, we'll see a little green signal here. And if there are A's, if there are DNA in other parts of the slide, and that DNA has a stretch of A's, well, we'll see things glowing there. And there are reasons why scientists want to do this. For instance, we talked about fluorescence in situ hybridization experiments where the scientists were inferring the genetic makeup of secondary oocytes from first polar bodies using fish, and there are millions of infinite number of other reasons why you would want to use this technology. Many different experiments you could use with this technology. But this is molecular hybridization, essentially just sticking, using a probe to search for DNA sequences, and it will identify those sequences by complementary base pairing and reforming that double-stranded DNA based on the complementarity between the probe and the DNA sequence. Okay, so that was questions 10, 21, 22, 23, and 24.